Welcome to this microlearning on antigen processing and presentation. In the next few minutes, we will unpack how cells convert proteins into signals the immune system can read, and why that matters for infections, cancer, autoimmunity, and vaccines. Settle in and let's translate complex cell biology into clear, practical insight. Here is our roadmap. We start with a quick overview, then meet the MHC molecules that display peptides. Next, we trace the processing pathways that generate those peptides before zooming in on how loading and quality control work. We close by linking these mechanisms to real clinical challenges and opportunities. Let's begin with the big picture. Antigen processing and presentation are the language immune cells use to talk. Cells break down proteins into peptides and display them on MHC molecules, allowing T cells to sense what is happening inside and outside a cell. This simple display system underlies adaptive immunity. Why present antigens at all? Converting proteins into peptide MHC complexes kickstarts adaptive responses, guiding T cells toward pathogens and tumors while sparing healthy tissue. Just as importantly, continuous presentation enables immune surveillance, so T cells can patrol for anything foreign or abnormal and respond before threats gain ground. Who presents antigens? Professional antigen-presenting cells, dendritic cells, macrophages, and B cells, capture, process, and display peptides to T cells. Dendritic cells are standout specialists at priming naive T cells, igniting robust responses. But presentation alone is not enough. Co-stimulatory signals provided by APCs act like a green light for T cell activation and differentiation. Now to the display platforms themselves, MHC molecules. Class 1 and Class 2 have distinct structures, sample different protein sources, and talk to different T cell partners. Understanding who presents what and to which T cell explains why some responses kill infected cells while others coordinate broader immune defenses. MHC Class I is built from a polymorphic heavy chain paired with beta-2 microglobulin. It samples intracellular proteins, typically presenting short peptides around 9 amino acids to CD8 cytotoxic T cells. Think of the image as a visual cue for precision and selection, echoing how tightly Class I controls its peptide cargo. MHC Class II is an alpha-beta heterodimer designed for longer exogenous peptides, often 12 to 20 residues. These complexes are recognized by CD4 helper T cells, which orchestrate responses by activating B cells, guiding macrophages, and shaping cytokine environments. Together, Class II displays the outside world so immunity stays coordinated and focused. With the cast introduced, let's trace the pathways that generate peptides. We will compare the endogenous route that feeds MHC-I, the exogenous route for MHC2, and the special cross-presentation pathway that lets dendritic cells prime CD8 responses from external antigens. Follow the flow as peptides travel to the surface. Endogenous processing starts in the cytosol. Proteins are chopped by the proteasome, and resulting peptides are ferried into the endoplasmic reticulum through the TAP transporter. There, ERAP trims them to fit just right, and they load onto nascent MHC-I with beta-2 microglobulin. Stable complexes then ride to the surface for CD8 T cell recognition. Exogenous processing focuses on what the cell takes in. Antigens are captured by endocytosis or phagocytosis and routed into increasingly acidic endosomes. Cathepsins cleave them into peptides, and in late endosomal compartments those peptides bind MHC2. The resulting complexes move to the cell surface, where CD4 T cells read the message and coordinate responses. Cross-presentation is the clever exception. Certain dendritic cells can divert engulfed proteins into the cytosol, let the proteasome process them, and load the resulting peptides onto MHC-I. This equips CD8 T cells to recognize antigens that were not produced inside the presenting cell, a key tactic for anti-tumor and antiviral priming. Having mapped the routes, we now focus on loading itself. Cells use rigorous quality control to ensure only the best-fitting peptides reach the surface. Think of it as a molecular editorial process. Chaperones stabilize empty MHC molecules, test peptide candidates, and release stable complexes that T cells can confidently interpret. For MHC-I, chaperones do the early heavy lifting. Calnexin, calreticulin, and ERP57 stabilize the empty molecule and help position it for peptide binding. Tapicin then acts like a proofreader, favoring higher affinity peptides and promoting stable complexes. The result is a sharp, reliable signal that efficiently activates CD8 T cells. For MHC2, an invariant chain blocks the groove during biosynthesis, and its clip fragment temporarily occupies the site. HLA-DM catalyzes the exchange, releasing clip and stabilizing the groove so high-affinity antigenic peptides can bind. Only then do stable peptide MHC2 complexes head to the surface for CD4 recognition. Let's connect mechanism to medicine. The way antigens are processed and presented shapes who escapes detection, who triggers autoimmunity, and how vaccines work. Small tweaks in these pathways can tilt the balance between protection and pathology, making them compelling targets for diagnostics, therapies, and next-generation vaccine design. Pathogens fight back. Some viruses reduce MHC-I expression or block TAP, blunting peptide supply and hiding from CD8 surveillance. These evasion strategies illustrate an evolutionary arms race. As hosts refine antigen presentation, pathogens evolve countermeasures, pushing immunologists to design therapies that restore visibility and reinstate T-cell pressure. When tolerance fails, presentation can turn on the self. Inefficient clearance of dying cells increases the chance that self-peptides enter the MHC2 pathway, breaking tolerance and fueling autoimmune disease. Post-translational modifications like citrullination create neoepitopes that class II presents, contributing to conditions such as rheumatoid arthritis and type 1 diabetes. Vaccine success relies on the right epitopes in the right context. Formulations select high-affinity peptides to drive strong T-cell activation and lasting memory. Adjuvants mature APCs to enhance processing and presentation, while nanoparticle delivery can promote cross-presentation, ensuring both CD4 and CD8 arms engage for comprehensive and durable protection. Thanks for joining this walkthrough of antigen processing and presentation. You now have a clear map from protein breakdown to T-cell activation, and how these steps influence infection control, cancer immunity, autoimmunity, and vaccine design. Keep exploring and apply these principles whenever you evaluate new immunology data or therapies.